Today, we're diving into a really fascinating, kind of radical new way to see the ghosts in the machine of life. I'm talking about all those hidden biological operators that our current tools just can't quite see.
We're going to take a trip into what you might call life's twilight zone. You know, that blurry, fuzzy boundary where our neat little definitions of what's alive totally break down. This is where you find the things that aren't exactly organisms, but they're definitely not just inert chunks of matter either. These are the hijackers, the phantoms, the things that work right on the edges of the big biological systems.
So here's the big question, right? How do you find something that's basically hiding in plain sight? It's a huge mystery. Some of the most influential players in all of biology are practically invisible to us, and it's because they just don't fit into our clean categories. They completely break our tools of classification. They are absolute masters of disguise. Just to give you a feel for it, let me introduce you to a few of these biological phantoms. First up, we've got the recently discovered obelisks. They're these super mysterious loops of RNA just hanging out in our gut. Then you have things called ghost archaea, which are actual cells with genomes smaller than some viruses. And then there are Trojan genes, tiny little hijacker genes that can literally rewire photosynthesis across entire oceans. It's wild stuff.
Okay, so why is it so hard to spot these things? Well, it's because our old traditional methods, they're just failing us. For decades, biology has been asking one main question: what is it made of? So we sequence genomes, we catalog all the parts. But for these phantoms, that's just not going to cut it. It's kind of like trying to understand a master key just by analyzing the metal it's made from. You're missing the whole point.
So if just looking at the key isn't enough, we need a completely new way of seeing, a whole new principle. What if instead of asking what something is, we start watching what it does when it's put under some kind of pressure?
And this, right here, is the crucial shift in thinking. It's a game changer. We stop asking, what is it made of? And we start asking a new question. How does it pass through a bottleneck? We stop studying the static thread, and instead we watch the dynamic, unique way it moves through the eye of a needle. We're going from taking a single photograph to watching the entire movie.
And that action, that journey through the bottleneck, creates something called a transition fingerprint. It's the unique, measurable signature that an entity leaves behind as it squeezes through. And that fingerprint, the shape of its journey, is often way, way more revealing than just a static photo of the thing itself. Now, this isn't just some vague, high-level idea. It's a really structured process. There's a whole pipeline. First, you find the system's critical bottlenecks. Step 2, you measure the trace it leaves as it passes through. Then, use some pretty clever math to boil that trace down into a compact fingerprint. And finally, you interpret that fingerprint to figure out what the entity is, and just as important, what it's doing. All right, so enough theory. Let's see this principle in action. We're going to look at 5 totally different examples that show how this idea can unlock mysteries across completely different scales of biology, from a single molecule all the way up to an entire ecosystem. Let's start at the absolute smallest scale, which is kind of the proof of concept for all of this. Scientists can actually identify a single protein, not by what it's made of, but by pulling it through a tiny microscopic hole, a nanopore. As it gets pulled through, the electrical current stutters in a very specific way. That stick-slip pattern is its transition fingerprint. It's a literal physical needle eye. Okay, now let's scale things up a bit, back to those mysterious obelisks in our gut. In this case, the bottleneck isn't a physical hole, it's an ecological one, like, say, a limited resource that all the microbes are competing for. When an obelisk is in the system, it changes the way the entire community interacts. Its transition fingerprint is the unique ripple effect it creates across that whole network. And what about that ghost archaeon, the one with the super tiny genome? Well, we can stop trying to define it by all the genes it's lost. Instead, let's look at the ultimate bottleneck for any cell: division. Its fingerprint is the essential network of genes that it kept, the absolute bare minimum wiring it needs to squeeze through that bottleneck and make a copy of itself. And the Trojan gene, that tiny little MBAA gene, sneaks into ocean bacteria and hijacks their photosynthetic machinery. That's a critical energy bottleneck. Its fingerprint is a measurable change in the flow of that energy. We don't even need to find the gene itself. We just have to see the unique shadow it casts on the system's power plant. And finally, let's go huge. Let's go geological. Ancient bacteria used to build these little internal chains of crystals to navigate, kind of like tiny compass needles. For them, the bottleneck was the Earth's entire magnetic field, and the fossilized crystal chains they left behind, those are a physical, materialized transition fingerprint, showing us exactly how they passed through that global field millions of years later. So, as you can probably tell, this is way more than just a collection of clever analogies. It's a formal scientific principle. It's like a universal lens that leads directly to predictions you can actually go out and test. And here's the core idea put a bit more formally. For these hidden, minimal, Trojan-like things, the way they pass through a bottleneck is a much better, more stable way to identify them than just looking at what they're made of. In short, the how is so much more important than the what. This principle also lets us do something really cool. It helps us tell the difference between a system that's just a little stressed out and one that's undergoing a fundamental change. Think about traffic. A simple traffic jam is just a slowdown, but the flow is still basically linear. But gridlock? That's totally different. The entire pattern, the topology of the system changes. New loops form, cars get stuck. A transition fingerprint can spot that fundamental shift. And look, this isn't just theory. This leads to concrete, falsifiable predictions that scientists can test right now. I mean, could we actually design a nanopore that spots those obelisks without ever having to do any sequencing? Could we analyze something like an EKG trace to detect a hidden infection before any symptoms show up? These are real, exciting possibilities that come directly out of this idea. And that brings us to what might be the most exciting implication of all. What if we can move from just observing these systems to maybe, just maybe, actively controlling them? This brings us to a concept called systemic acupuncture. It's a great name for it. The idea is, if bottlenecks are the absolute best places to listen to a system, well, then they might also be the best places to apply a tiny, precise push. By intervening at just the right point, the needle eye, you could potentially steer the entire system with very little effort. And that leaves us with a really provocative final thought. If these bottlenecks really are levers for control, what systems could we start to steer? An entire city's traffic grid, our own metabolism. The needle eye principle doesn't just give us a powerful new way to see the world, it might just give us a new way to help shape it.